Hi, I'm Lauren.
I'm Isabella.
I'm Natalie,
And I'm Jalissa.
and today we're going to be going over grave case study number 3. Specifically, we're going over the genomics of parallel experimental evolution in Drosophila.
The first question we aim to answer was what was the main research question in the study?
The main research question in the study was what are the genomic foundations of adaptation in sexual populations?
Adding to that, the primary goal of the experiment was to establish whether emerging contrast between asexual and sexual populations is sustained over a wide range of evolutionary durations in
Yes,
sexual populations.
and the second goal is to determine the degree of which phenotypic and genomic parallelism occurs between populations that share recently imposed versus long sustained selection regimens. And specifically, the study is testing whether different Drosophila populations that are in the same selection regimen will go through parallel evolution.
The third goal was to probe the evolutionary genetic mechanisms underlying parallelism within each set of 10 populations that share a recent selection regimen.
The
Okay.
second question is what were the main results of this study?
The first thing that they went over was phenotypes, and they found out that newly derived NCO populations had superior reproductive output compared to long-standing CO populations during a single time interval. And the overall fitness character results show a lack of evolutionary differentiation between long-standing and newly derived populations sharing the same distal selection regime.
And so the second part was looking at the genomic analysis, and the results that were obtained was, well, first they focused on 3 types of variation in the pooled population sequence. Specifically, they look at single nucleotide polymorphism, also known as SNPs. They looked at the structural variants and the transposable elements. And so overall, the results showed that adaptation in experimentally evolved populations is driven by selection and standing genetic variation. Overall, the populations with the same selection regimen had no significantly differentiated SNPs. Populations with different regimens showed hundreds to thousands of differentiated SNPs.
The final main result was in heterozygosity. There was no evidence for widespread reductions in SNP heterozygosity across our 100 kb windows. There are a number of local depressions in heterozygosity in all of our populations. Overall, A-type populations had the lowest heterozygosity, C-type populations had the highest heterozygosity, and there were not huge regions of extremely low diversity.
The third question is what were the main conclusions of this study?
So the main conclusion was that experimental evolution of moderately outbred Drosophila involves reproducible and extensive changes across the fruit fly genome.
And so the other main conclusions was that marked functional and genomic differentiation between newly derived populations that do not share recent selection regimens is comparable to that found between long-standing populations that do not share selection regimens, despite large disparities in numbers of generations under divergent selection. And this finding specifically supports the contention that convergent or parallel depending on favored usage, evolution can result from polymorphism that were shared in an ancestral population, as all selected lines in the study were derived from a common ancestral population.
The scale of our study underscores the following conclusions about the evolution of outbred sexual populations. Number 1, their evolution can be fast. Number 2, their evolution can be repeatable from nucleotide to fitness thanks to standing genetic variation. And number 3, their adaptation does not wait for new functional mutations and subsequent hard selective sweeps.
Furthermore, the results of the study indicated that most of the genomic sites under selection in our study are undergoing a shift between stable balanced polymorphism, not selective sweeps towards fixation of a single genome-wide haplotype. And the study is not intended to directly model what occurs in a natural population. Instead, the study is intended to use extreme and unusual evolutionary histories to test competing evolutionary genetic hypotheses.
That concludes our conversation and our discussion. Thank you.
Thank you.
Thank you.